You can.
Good. So today I want to talk to you about microbial model communities and how they reveal widespread metabolic interdependencies. And I want to start with a broad introduction. So I hope until now you have already seen in class that bacteria drive biogeochemical cycles on Earth. And it's not one superbacteria that is doing this driving of biochemical cycles, but it's actually a multitude of diverse bacteria doing it. And you see in these very simplified diagrams, the carbon cycle and the nitrogen cycle that are, in this case, represented in the ocean. And these steps are now being shown, are being carried by different types of bacteria. And as I said, this is very simplified. So different types of bacteria are connected through different steps in the biochemical cycles. Now, adding to this, bacteria are also connected by metabolic dependencies. And in this very simple schematic, I show how this bacteria is producing essential metabolites that this other bacteria cannot produce for itself, instead needs to consume them from another bacteria that produces it. So this type of metabolic dependencies create this type of networks of interactions. This is also simplified, but I want to show you how it would more look like in nature. So this is a network of co-occurrences, and co-occurrences don't immediately equate to interactions, but it gives us an indication of bacteria that occur in the same location at the same time. And you see that in gray. This whole cluster here is bacteria that are happening at the same time in the same location in Lake Erken. This network was constructed with data collected over 8 years of a time series. So the question I have for this network is how do we start to study these very complex microbial interactions that arise from this interconnectedness of biochemical cycles and metabolic dependencies? Now, to even start trying to answer this question, I want to present to you something that you have probably learned before, which is the biological organization of life and how we have been studying life in the past. So this was published in 1962. And it shows how hierarchically life organizes from the cell to the tissue and the organ, and then organs into organisms, populations, and communities. And this has been visualized for decades for larger organisms. And I've been questioning why haven't we started visualizing this also for microorganisms? And so this is my first visualization on an illustration made by a colleague, Sergio Tuso. And you see, drawing parallels to this biological organization of life, where we have cells, then populations, as in difference to the big biological organization of life, populations come next, and then interspecies interactions, functional consortiums, and microbial cohorts. And these names I gave them. In the literature, there have been kinds of names given, but there's very chaotic, many different types of names given. So this is a proposal I have, and we have submitted this to the ISME journal. So crossing finger for good and positive reviews. And this is just the conceptualization of how I think we can study this type of interactions, because as you maybe know, right, when we study the properties of biological organisms, right, the way we study cells is different to the way we study tissues, and what tissues tells us, right, is different to what organs tells us, for example. So it's the same in microbes. And when I try to bring these concepts now to methods, I want to bring you down to these three levels where we have environmental communities, we have individuals, and I propose forward what I call model communities. Model communities are a dilution from the environment, so cells that were normally co-occurring in the environment that get diluted and then sorted randomly into containers to then try to cultivate them together into what I call a model community. With this then levels of methods, we can start looking at different levels of interaction, diversity, but also physiological resolution. So when we bring these different levels, for example, for DNA extraction, we can start looking at metagenomics, or if we grow these individuals into cultures, we can look at genomes from pure cultures, or these individuals, we can look at the level of single cell genomics. And if we want to look at metabolic activity, we could also extract the metabolites and look at metabolomic studies, for example. Now, I want to give you some examples. So when we overlap the methods with the concepts and we look at individuals, I want to talk how pure cultures differ greatly from wild microbes. And so what we have learned from in microbiology through decades has been a lot of it thanks to pure cultures. And it has been a lot what we have been learning. So in 2022, my group published a collection of genomes, including genomes from isolates, and all of the genomes from environments that were in a public database that came from aquatic environments, host-associated environments, and terrestrial environments. And in total, it was about 50,000 genomes that when they replicated, there was around 25,000 species. And what you can see is that the species that were isolated were very different to the species that are found in the environment. So from this, we get that about 3% of the species of the environment had culture representatives. And this is relevant not only in the context of how different they are, but for example, when we look at genomic features such as the genome size, we start looking at how different their genome size are as well. So you see in a density plot, the genome size of the isolates has an average of about 4 megabase pairs, and this is actually the genome size of E. coli, which is the most studied microbe. But the genome size of aquatic microbes has an average of 2 megabase pairs. And for prokaryotes, bacteria in general, genome size directly correlates with number of proteins. And so the bigger the genome size, the more functions the genome carries. And so this is already showing us how, even though we have learned so much about microbes from isolates, we're also biasing our view of what microbes are doing and how they're behaving if everything we're learning from microbes is based on pure cultures. And so the other point of view on pure cultures is when we're trying to understand interspecies interactions and we are looking at combinations of two pure cultures growing together, we start to understand, are they competing? Are they mutualist? Or do they have neutral relationships, right? And this study published in 2023 has, for example, when there's co-cultures between organisms that do not produce all the essential metabolites called auxotrophs and prototrophs, right? This is the picture that emerges. There is a lot of mutualism happening and about 25% of maybe negative interactions. Now, when this is compared, for example, with cultures from kefir, grown in plates or in liquid culture, the same pairs show very different picture. So when they are grown in liquid culture, more mutualisms emerge. And when they are grown in plates, more negative interactions emerge. So studying interactions with pure cultures under specific conditions can show us also biased pictures of how interactions are looking like, depending on how we are looking at these interactions. So this is just a comment on studying interactions at this level of life organization. And so with this in mind, I want to then go into my favorite way of looking at interactions, which is model communities. And I want to spend the rest of my talk going over work that was done by PhD student Armando Pacheco and PhD student Anna Couch um, on a sample that was taken on the Baltic Sea. So this sample was taken in 2021, and we set up model communities from there. And as I said, this model community setup is taking samples from the environment and then doing dilutions so that random organisms end up together with other organisms that co-occurred in the same sample in same wells. So that's what I try to represent here. And then after the inoculation, we incubated for 4 weeks, and we tried to give them temperature and light regime as close as possible to natural conditions. And the media we used is filtered water from the same environment they came from, so they would have nutrients as similar to what they were coming from. So with this method, we were inoculating 8 different 96 well plates, and we were trying to inoculate some plates where we had 2 cells per well, some plates where we had 8 cells per well, 15 cells per well, and so until we got one with 100 cells per well. And I'm going to show you in the next plot the results, the first results. So in the x-axis, you see how many cells per well we inoculated. And in the y-axis, you see out of the 96 well plate, how many wells were positive in growth as measured by obtaining genomes from the metagenomes we sequence out of those cultures. So what you can clearly see is the higher number of inoculated cells leads to the higher cultivation success. And I want to pair this up with another plot where you see in the x-axis the number of inoculated cells, and in the y-axis you see how many different species we found growing together in the same culture. And you also see, in addition, this other plot where we have not 96 well plates, but bottles where we inoculated more cells. And you see the more cells we inoculated together, the more species we found growing together at the end of the experiment. So in total, in this experiment, we have 200 model communities that led to successful co-growth. I think 90 of them were individual species growing together, but 110 were co-growth of organisms that came from the same sample. And so in the following plots, I'm going to show you what we found on these model communities. So the next table I'm showing you, it has a lot of lines, but I'm going to slowly walk you through. So it's a heat map where you see for the model communities where we only found one organism growing, one species, or 2 species growing together, or 3. So if it's gray, it's positive, and if it's white, it's negative. And then each line is a species that we found growing in these cultures. And so if the species has a white spot, that means the organism we found growing hasn't been described by pure culture ever, or hasn't ever been sequenced and described by metagenome assembled genomes from the environment. So what you can see first in this figure is that 70% of the species that we were able to grow in our experiment have never been described before. And this is, I think, very exciting, because it's showing that with this method that we are using in my lab, we're able to cultivate organisms that were previously uncultivated and undescribed. The next thing that I want to highlight in this table is that with green, we highlighted the organisms that only grow when there's other organisms around. And with this orange, we highlighted the ones that were only found growing on their own. The ones that have no color highlight, they grow irrespective if they're alone or if they're in groups. Okay? So what you see is that most of the organisms that we found growing in this type of cultivation only grow when they're growing in groups. And this is already, I think, very important because it shows how many of the organisms out there in nature really need others around them in order to be able to successfully grow. And in the next plot, I want to show you our ideas of why we think they needing each other. And so this is looking at the biosynthesis of essential metabolites such as amino acids and vitamins. And in this plot, you're going to see all the organisms that were found growing only on their own. They're in orange, and each column is the genome of one organism. In green, you're going to see the ones that were growing in groups of 2 or in groups of 3 or in groups larger than 3. Each row is the pathway for either an amino acid in the part A or a vitamin in the part C. And if the color is very strong, that means that genome had the full pathway in its genome to produce this essential metabolite. But if the color is very white, that means it did not have the pathway. Okay. So I think this heat map on its own, maybe it's like not that informative. So we summarize this data into a box plot where you can like see the summary of this plot. And what you see is that for amino acids, organisms that grow on their own, in average, have the potential to produce all of the amino acids. Also when they grow in pairs, right? There's little exceptions. But when they grow in groups of 3 or more than 3, in average, they actually produce less and less of the amino acids for themselves. And for vitamins, the picture looks similar but also different because when they grow on their own, in average, they can produce 75% of the production of vitamins. And as they grow together with more organisms, this percentage is dropping. Now, amino acids are building blocks of the cell, right? And I think this is why it needs to be complete if you're growing on your own, whereas vitamins are recyclable. They are not like a building block. And because the media we provided is filtered water where they came from, there was probably residual vitamins in that water. And this is probably why they were getting away with not having full pathways if they were growing on their own. Now I want to show for the organisms that were growing in groups more than 3, I want to show the next plot where you'll see in each of the columns, it's one of the steps of the biosynthesis of, for example, here's the arginine. This is chorismate, which is a precursor of an amino acid. Cysteine, histidine, isoleucine, and so on. These are all the amino acids, and these are the vitamins. And each row is one of the model communities. So what you see when it's very dark is all of the genomes in that model community had the step to produce that amino acid. If it's completely white, is none of the genomes had the step to produce the amino acid. But what you can see in this figure is that when you look at the overall picture, there is really not white, but light colors in some of the steps, meaning together the members of the community were able to produce the amino acids, right? Maybe individual members didn't have the potential to produce the amino acids, but when combined, at least one of the members had each of the steps. There was, of course, some steps that all of them had, but all of the steps were had at least by one of the members of the community. And this is why I believe we were able to cultivate organisms that wouldn't grow otherwise on their own. So I want to end by showing you of the organisms that we were able to grow in these model communities. We were in total growing 71 species. When we did the overall environmental survey, we found 701 species in the Baltic Sea. So the big circle is all the species we found in the environment, and the small circle is the ones we were cultivating. So overall, we were only managing to cultivate a small percentage of what's in the environment. And even the ones that we didn't find in the environment by metagenome assemble binning, we were still finding them by recruitment. So they are there, they're just less abundant. Nevertheless, these ones that were found both by metagenomic approaches and cultivation approach are very abundant in the environment. And this I want to show you in this figure. So in the y-axis, you see the relative abundance. And in the x-axis, you see the average pathway completeness of both amino acids and vitamins combined. And so you see, when it's a 1, it means it can produce both amino acids and vitamins. So we put them together. And 0 would mean it can produce none of them. And there was a group of organisms in the environment that seemed to be able to produce none of that. And we didn't manage to cultivate those with our approach. So it would be interesting to figure out how to cultivate those, right? But of the most abundant ones, you see the purple ones we were able to cultivate with our method. We were able to cultivate a lot of the most abundant. And we were also able to cultivate in red some of the least abundant from the environment. And so with that, I end my talk for today and I open the floor to questions. I want to acknowledge my research group and you for your attention.
Thank you. Thank you so much for the